Hi everyone, and welcome to our video on multiple alleles. This is again part of our module 6 work, and what we're going to be doing in this video is having a look at specification reference 6.1.2b part 1, genetic diagrams to show patterns of inheritance, with that focus on the multiple alleles. So before we can really delve into any of these diagrams that we're going to have to be able to do, we need to understand what on earth we're talking about when we use this phrase multiple alleles. Now, quite simply, what we're looking for here is a characteristic where there are 3 or more alleles for it in the gene pool of that population. So obviously we've had a look in our monohybrid inheritance, for example, at characteristics where there are literally 2 options. Here, we're now looking at a characteristic where we've got at least 3 alleles for it in that total gene pool of the population. What we'll do then is we can actually have a look at a couple of examples, because it's actually easier to get your head around this when you can physically see the examples. So the first example we're going to have a little look at is human blood group. Hopefully we do know that there are a grand total of 4 blood groups as far as humans go, ignoring rhesus factors, etc., which is A, B, AB, or O. So if you were to look at your blood group, you would be one of those 4, okay? Now, obviously we're not going into the realms of the positives and the negatives that you also get with your blood. We're just focused on whether you're A, B, AB, or O. So this obviously meets our criteria for multiple alleles, because we don't have just 2, we have obviously 3 or more options. And what we can see here is that we've got 3 alleles for it. What we need to understand then is how these different alleles are going to interact to give us that final blood group. Now, as far as human blood group goes, then we see 2 patterns of inheritance here. The first one is dominance, so just looking to see if this trait is present along with this one, then this one is dominant to it, so we see that characteristic. But we also have this thing called codominance. Now, codominance is new for us at this point, which just means that if we've got codominant alleles present within the genotype, then we get both of them contributing to the phenotype. So we have these 2 different patterns of inheritance we need to be aware of: dominance and codominance. So if we have a little look then at how this all works. Basically, those 4 blood groups, of which you will belong to one, are going to be determined by 3 alleles. Now, these 3 alleles exist for this isoagglutinogen gene, okay, which we just term capital I. And we find that on chromosome 9. So that would be its gene locus, is that specific position on chromosome 9. And it's going to be given this capital I as the first bit of its notation. When we then want to work out which of the 3 alleles we've got these 3 ways to write it: IA, IB, IO. Do notice the A, the B and the O are all in that superscript position to the right of the capital I. So I obviously stands for the isoagglutinogen, and then ABO is the actual allele that is present. What we can see in this little statement at the bottom is the dominant statement. So IA and IB are dominant to IO. So what that means is that if we've got either IA or IB present along with an IO, then we will see either the A or the B, not the O, because the O is not dominant to any of them. Okay, so A and B are dominant to IO. We also have a second statement here that IA and IB are codominant. So what we can see by this, remember our definition of codominance is that if those 2 alleles are present, they both contribute to the phenotype. So this, if we think back to our blood groups, if we happen to have IA and IB present, then that's going to give us that AB blood group. So we've obviously got IA, we've got IB, they're both contributing to that final phenotype. So when it comes to the actual genotypes and the phenotype that we then get. This little table at the top just summarises the different combinations of genotypes and their corresponding phenotype. So we can see, first of all, if you've got both IAs, then clearly blood group is going to be A. If we have IA and IO, then IA is dominant to IO, so we're still group A. Group B is very similar. IB and IB makes group B, and IB-IO makes group B because IB is dominant to IO. Then when we come on to our third row here, we've got IA and IB. They demonstrate codominance, remember, so this is our codominance. And as a result of that, we get group AB. They're both shown in that phenotype. The only way we get our group O is if you've inherited both IOs. So we can see there that the last blood group there, group O, is only if you've got both of your alleles as IO. Just for interest, you don't need to remember this for your exams. They're not going to literally say what percentage of the population of group B blood, but it's just an interest point really, that your group A equates to 38% of the population, your group B 10%, your group AB 3%, they're your rare people, and then your group O is the 48%. And remember earlier on we talked about this rhesus factor, the positive and the negative. So in brackets it just shows you the percentages of the population that would have that particular blood group with that rhesus factor. So from that you can see instantly that if you're AB negative, then you are a super rare little individual because you are one of only 1% of the population with that particular blood group. So generally if you're one of those people, always good to donate blood if you can. So, what kinds of questions could we see with this? Here's an example. Work out the blood groups of the potential offspring for a male of blood group A and a female of blood group B. And it tells us that both parents are heterozygous at the gene locus. So hopefully we remember that heterozygous then tells us that these are going to be different. Now, the first thing we've got to do is work out the genotypes of those two parents. So if we've got the male parent is blood group A and they're heterozygous, so they're different, we know we've got to have IA, and the only way they can be blood group A with different alleles present is if the other one is IO. Same thing for the female, we need to look at that first of all. So if we look at our female, blood group is B, and therefore heterozygous still, so we must have IB, and therefore the other one must be IO. So we've now worked out the genotypes of those two parents. Next thing to do then is to actually set out our little Punnett square. So really straightforward little Punnett square that we can do as usual. Do remember that when we are obviously setting out the Punnett square, our rules for where the male and where the female go. So females always going down and the males always going across. So we would have IA, IO in our little gametes, which we can stick in our circles as usual. And then the female is IB and IO. So then we just do the standard Punnett square, whatever is at the top, we stick in the two boxes beneath. So these two are going to be IOs. Whatever is on the left, stick in the two boxes to the right. So these two would be IBs, and at the bottom we'd have IOs. Then we just need to work out the potential phenotypes. So remember our rules. Let's start with our dominance rule. If we've got either IA or IB present with IO, then they are dominant to the IO. So this means our top right box, this one is going to be our type B blood, and then at the bottom left we've got type A blood because they're demonstrating dominance. Obviously the bottom right we're going to have type O because they're the only alleles present, and then remember A and B are co-dominant to each other, so that means that we have type AB blood in the top left box. So in fact these two parents could have offspring with every blood type imaginable, bless them. The other example we're going to have a little look at then are rabbits. Now rabbits have four coat colours. they've got agouti, which is this one in the bottom left here. So the hair basically has a grey base, yellow band and a black tip, which gives them that brownish looking colour. We then have the albinos, which are white-haired because they do not have a pigment. We then get the silvery grey ones, which are the chinchillas. And then finally on the right, the Himalayan, which is where they're white with those black feet, ears, tail and nose. Now, in order to work out what colour coat our rabbit will have then, we've got one gene with 4 alleles, and they've got this dominance hierarchy. So working out which allele is dominant to which other ones. So, first thing, our agouti is dominant to all alleles. So that's just given the capital C, nothing else there. Chinchilla, which is our CH in the superscript position, is dominant to the Himalayan, CH. And the albino, lowercase c, recessive to everything. Now, you're not going to be expected to remember these kind of dominance hierarchies. You don't have to memorize any specific examples here. You would get the information in the question to be able to solve any of these. So, let's have a little look then. What we're going to do is have a look at our first example, an albino rabbit being crossed with a heterozygous Himalayan rabbit. So if we start off with an albino, first of all, so our albino rabbit then quite simply is going to be both lowercase c's, because remember that's our recessive, it's the lowest on our dominance hierarchy. We're then going to be crossing that with our Himalayan, and this Himalayan is heterozygous, which means different alleles. So that means we must have the Himalayan, which is the C with a little H in the top right subscript position. And the only way we could have that is if the other allele was the lowercase c, because obviously Himalayan comes third in our hierarchy as we just saw. So what we're then going to do is obviously we are just going to set out our little Punnett square. Now in this case we haven't actually got any reference to which one is male and which one is female, so we're just going to put the albino at the top and then let's put our Himalayan at the side. So again we just fill these out, so little albinos first, oh dear me, little albinos, and then we can do our Himalayan, and then all we've got to do is identify the phenotypes. So if we've got both lowercase c's, these are going to be albino, and at the top we've got a lowercase c alongside the capital C with the Himalayan, and therefore they would be Himalayan. So what we can see here is that we've got a 2:2 ratio, so that is going to be the albino to the Himalayan. We'll just have one more go at an example here just so you can see how this dominance hierarchy works in one more case. So we've got a heterozygous agouti individual with a chinchilla allele, and they're being crossed with an albino. So we already know that if it's an albino, both lowercase c's. Then we've got our agouti individual, remember. Now if it's agouti, that means we've got to have a capital C, and the other one it tells us is a chinchilla allele. So that one is going to be a CH. Same thing as before, we've got no reference to which is male, which is female, so quick sketch out of our Punnett square. Let's put the albino at the top again, and then we've got a capital C and our chinchilla. So whatever's at the top goes into the 2 boxes beneath, whatever is on the left goes in the 2 boxes to the right, and then we just need to work out our phenotypes. So first thing, in our top 2 boxes we've got the same combination of alleles, one lowercase c, one capital C with no other notation, therefore they are going to be agoutis, because our agouti allele, remember, is dominant to all others. Then we've obviously got recessive alleles, and that goes against the chinchilla, so in both of these cases they are going to be chinchillas. So again we've got a 2 to do, and this one is going to be agouti to chinchilla. As always, I hope you found this video useful, so don't forget to subscribe so you can see when more videos are uploaded, and of course head on over to the website where you can find more useful resources. And don't forget, on this one, it's not about the specific examples, it's more about the technique. You don't have to memorise the dominance hierarchy for rabbits. You will not be asked to recall that. You will, however, potentially anyway, be asked to apply it to a situation given on your exam paper.